There's a moment, you've probably felt it, where the day doesn't end so much as loosen. Not sleep yet, something before sleep, a kind of giving up that isn't defeat. Your shoulders drop without deciding to. The grip you keep on your own thoughts goes slack and you don't notice it happening until it's already happened. That loosening isn't accidental. It has a biological address, a reason that lives in tissue, not in mood. Something is being removed from your brain right now. Not dramatically, nothing dramatic happens here. But while you lie still, while your breathing settles into that slower, almost embarrassing rhythm, embarrassing because it's so animal, so unguarded, a system that spent most of your waking hours waiting for this exact moment has begun to move. Welcome to the Sleepy Biologist. If you've been here before, you know the pace we keep, and if tonight is your first night here, I'm glad the channel found you when it did. This space exists because enough people wanted somewhere quiet to take biology seriously, without the urgency, without the quiz. Channel members keep that possible, and I don't say that lightly. There's a warmth to knowing this is, for some of you, a genuine nightly ritual. If you feel like leaving a comment before sleep takes you, even just a word, I read them. All of them, now. Back to what's happening behind your eyes. The brain is not a sealed container. That's the first thing. It seems sealed, protected inside bone, wrapped in 3 layers of membrane, kept behind one of the most selective barriers in the body. But it has a drainage problem. Or rather, it solved a drainage problem in a way that took neuroscientists embarrassingly long to fully see. Every organ that works hard produces waste. That's not a judgment, just chemistry. Muscle cells working hard leave behind lactate. Liver cells processing toxins produce compounds that need clearing. The brain, burning through roughly 1/5 of your body's entire energy supply, despite being maybe 2% of your body weight, produces waste at a rate that would be unsustainable without a dedicated removal system. For a long time, scientists assumed the brain handled this the way lymphatic drainage handles it everywhere else in the body. Lymph vessels collect interstitial waste, move it toward lymph nodes, process it, return cleaned fluid. Elegant, reliable, except, and this is the strange part, the brain doesn't have lymphatic vessels inside it, not in the tissue itself. The central nervous system is, in that specific sense, cut off from the body's main waste-clearing infrastructure. So how does the waste leave? That question sat, half-answered for decades. The answer, when it came, arrived through mice. Not a satisfying origin story, maybe. But most of the real ones aren't. A neuroscientist named Maiken Nedergaard and her team at the University of Rochester published work in 2013 that described, for the first time with clarity, a system that had been operating inside every human brain, every night, without anyone quite understanding what it was doing or how. They called it the glymphatic system. The name is a hybrid, glial cells and lymphatic function pressed into a single word. It isn't elegant, but the system it describes is something else entirely. Here's what they found. Your brain is surrounded and threaded through by cerebrospinal fluid, a clear, almost colourless liquid that cushions the brain inside the skull, moves through the ventricles, circulates through the subarachnoid space just outside the brain tissue. You produce it constantly. You absorb it constantly. It moves. Welcome to the Sleepy Biologist. If you've been here before, you know the pace we keep. And if tonight is your first night here, I'm glad the channel found you when it did. This space exists because enough people wanted somewhere quiet to take biology seriously, without the urgency, without the quiz. Channel members keep that possible, and I don't say that lightly. There's a warmth to knowing this is, for some of you, a genuine nightly ritual. If you feel like leaving a comment before sleep takes you, even just a word, I read them, all of them. Now, back to what's happening behind your eyes. The brain is not a sealed container. That's the first thing. It seems sealed, protected inside bone, wrapped in 3 layers of membrane, kept behind one of the most selective barriers in the body. But it has a drainage problem, or rather, it solved a drainage problem in a way that took neuroscientists embarrassingly long to fully see. Every organ that works hard produces waste. That's not a judgment, just chemistry. Muscle cells working hard leave behind lactate. Liver cells processing toxins produce compounds that need clearing. The brain, burning through roughly 1/5 of your body's entire energy supply, despite being maybe 2% of your body weight, produces waste at a rate that would be unsustainable without a dedicated removal system. For a long time, scientists assumed the brain handled this the way lymphatic drainage handles it everywhere else in the body. Lymph vessels collect interstitial waste, move it toward lymph nodes, process it, return cleaned fluid. Elegant, reliable, except, and this is the strange part, the brain doesn't have lymphatic vessels inside it, not in the tissue itself. The central nervous system is, in that specific sense, cut off from the body's main waste-clearing infrastructure. So how does the waste leave? That question sat, half-answered, for decades. The answer, when it came, arrived through mice. Not a satisfying origin story, maybe, but most of the real ones aren't. A neuroscientist named Micah Nedergaard and her team at the University of Rochester published work in 2013 that described, for the first time with clarity, a system that had been operating inside every human brain, every night, without anyone quite understanding what it was doing or how. They called it the glymphatic system. The name is a hybrid, glial cells and lymphatic function pressed into a single word. It isn't elegant, but the system it describes is something else entirely. Here's what they found. Your brain is surrounded and threaded through by cerebrospinal fluid, a clear, almost colourless liquid that cushions the brain inside the skull, moves through the ventricles, circulates through the subarachnoid space, just outside the brain tissue. You produce it constantly, you absorb it constantly, it moves. What wasn't understood was that this fluid also moves into the brain, not just around it. It enters along channels that wrap around the outside of blood vessels like a sleeve, follows those vessels as they dive deeper and deeper into brain tissue, and in doing so pushes through the narrow spaces between brain cells. It acts essentially as a flushing mechanism. Fluid in, fluid through, waste picked up, fluid out, and draining eventually into lymphatic vessels that sit along the dural sinuses at the edges of the brain, and from there rejoining the body's broader lymphatic system. The pipes exist, they just took an unexpected route. But here's what matters for tonight. This system, this slow, deliberate flushing, is not running at full capacity right now if you're awake. It throttles. It runs at a reduced rate during waking hours, partly because the brain's own activity gets in the way, partly because of something subtle that happens to the actual physical structure of brain tissue when you fall asleep. The spaces between your brain cells get bigger, not permanently, not dangerously, but measurably, by something close to 60% according to some estimates, though those numbers were derived from mouse tissue and the precise figure in humans is still being worked out. The cells themselves don't shrink. What changes is how much fluid can move between them, like the difference between water moving through gravel and water moving through packed sand. Sleep opens the gravel, and the glymphatic system already waiting surges. There is something that feels almost too tidy about this, and I want to sit with that discomfort for a second, because science that seems too clean sometimes is. The glymphatic story has critics. Some researchers have questioned the exact mechanics of fluid movement, argued that the pressure gradients involved don't fully support the flush model as described. The system exists, that isn't in dispute, but how it works at the fine scale, how exactly the fluid moves, what drives the flow beyond simple pressure, those questions aren't entirely settled. I find it more interesting, not less, for that reason. It means there's something real happening inside you right now that we can describe but can't yet fully explain. That's not a gap in the story. That's the story. What gets removed matters. Not in an abstract way, in a very particular, very chemical way. One of the primary targets of glymphatic clearance is a protein called amyloid beta. You may have heard of it. It accumulates in the brains of people with Alzheimer's disease, clumping into plaques that disrupt the way neurons communicate. The connection between amyloid beta and Alzheimer's is complicated and still debated, but what isn't debated is this. Amyloid beta is produced continuously during normal brain activity, and it accumulates in the interstitial spaces between neurons, and during sleep its clearance rate increases substantially. Your brain is, in a very literal sense, cleaning out a protein that, if it stays, becomes part of a pathology. This isn't alarming, or it isn't meant to be. It's the opposite of alarming, actually. It's evidence that the clearing process works, that it happens every night, that sleep isn't passive in any sense that deserves the word. What happens while you lie still and stop being aware of yourself is as active as almost anything your brain does with your eyes open. More active in some ways. There's a low hum coming from somewhere in the house. You may not notice it. The kind of sound that only exists in the pause between thoughts, the refrigerator maybe, or something in the walls. It sits there underneath everything, constant and easy to miss. The glymphatic system works something like that. Constant, easy to miss. Present in every night of your life without once asking you to pay attention to it. You probably haven't thought about it before tonight, and it hasn't needed you to. One of the mechanisms behind the surge in glymphatic activity during sleep involves a particular kind of brain cell, not a neuron. We'll spend more time with these cells later, but for now, they have a role in managing the channels through which cerebrospinal fluid enters brain tissue, and when their behavior changes during sleep, the channels respond. The fluid moves differently, more of it gets through. The cells are called astrocytes. They're one kind of glial cell, and the word glial again comes from the Greek for glue. Supportive cells, filler cells, background cells, that's what scientists used to think. What they actually do is one of the stranger and more humbling revisions in modern neuroscience, but that's a chapter of its own. Right now, in the quiet, your cerebrospinal fluid is pressing gently through tissue, not rushing, not dramatic in any way. The movement is slow, viscous fluid through narrow channels, measured in micrometers per second, and it's carrying things out, metabolic debris, proteins, the chemical residue of a day's worth of thinking and perceiving and holding things together. You don't feel it, you won't remember it. But by morning the tissue will be different for it, not cleaned in some absolute sense. Biology doesn't deal in absolutes, but changed. Shifted toward a state where the neurons inside it can work again with something closer to their full range. Something is being removed. It has been removing quietly since you stopped fighting the weight behind your eyes, and the cells doing the removing are the ones guiding the fluid, managing the spaces, holding the channels open. They were there all along. You just never had a reason to think about them before. It doesn't announce itself. That's the first thing worth noticing. Most of what your body does comes with some signal. Hunger arrives as a clench, fatigue as a pulling weight, thirst as a dryness that sits in the back of the throat and refuses to be ignored. but cerebrospinal fluid gives you nothing. No sensation, no signal. It moves through you in complete silence, and the fact that you've never felt it doesn't mean it isn't doing something. It's doing quite a lot, actually. The fluid itself is strange to describe. Clear, colourless, or nearly, sometimes faintly yellowish if something's wrong, but normally clear, slightly salty, lighter than blood, thinner than you'd expect something so important to be. About 150 millilitres of it in your body at any given moment, which is roughly the volume of a small juice glass. Not much, and yet the brain and spinal cord float in it, are cushioned by it, are in some ways entirely dependent on it for survival. You're producing it right now, constantly. Around 500 millilitres every day, which means the entire volume turns over roughly 3 times in a 24-hour period. It doesn't pool, it circulates. The main site of production is a structure inside the brain called the choroid plexus, a tangle of specialized cells and blood vessels that sits inside brain's ventricles. The ventricles are cavities, hollow chambers inside the brain tissue itself. 4 of them, connected, forming a system that the cerebrospinal fluid moves through before spilling out into the subarachnoid space. The gap between the innermost and middle layers of the membrane wrapping around the brain and spinal cord. From there it moves downward, surrounding the spinal cord before being slowly reabsorbed. That's the broad loop. In and out, continuously your whole life. But there's a rhythm layered inside that loop that most descriptions leave out. I think about tides when I try to understand this. Not ocean tides, nothing that too dramatic. Something smaller. The way water moves in a very shallow estuary, barely perceptible, following something you can't directly see. There's a pulsation to cerebrospinal fluid movement that follows the heartbeat. The brain expands very slightly with each pulse of blood, and that expansion nudges the surrounding fluid. A tiny displacement, rhythmic, constant. And then there's a slower rhythm, breathing. Each inhale and exhale changes the pressure inside the chest, which changes the pressure in the spinal canal, which moves the fluid a little further, a little differently. The fluid is, in this way, caught between 2 rhythms it didn't create, pushed by the heartbeat, nudged by the breath. Neither of those systems knows it's coordinating the other. What changes at night is harder to pin down precisely, and I want to be honest about that because the science here is still being worked out. What we know is this: lymphatic activity, the movement of cerebrospinal fluid into and through brain tissue along those perivascular channels described in the last chapter, increases substantially during sleep, particularly during slow-wave sleep. The fluid that has been circulating at the brain's edges, moving through the ventricles and the subarachnoid space, begins penetrating more deeply. The channels open, or rather, they were always open, they widen, and the fluid follows. Something about that deserves a moment. The channels aren't manufactured fresh each night. They exist all day, running alongside blood vessels as they dive into brain tissue, lined by the end feet of astrocyte cells that press against the vessel walls and create a kind of guided path for fluid movement. During waking hours, fluid moves through them, but slowly and not very far. The tissue doesn't allow it. The cells are packed more tightly, the spaces between them narrower, the resistance higher. Sleep relaxes something, not metaphorically, physically. At the cellular level, the interstitial space, the gap between brain cells, expands. The mechanism isn't fully understood, but it involves ion transport, and it involves what neurons stop doing when they quiet. The tissue essentially loosens its grip on itself, and the fluid presses through. There's a smell in the air tonight, Something faintly green, like rain on dry concrete, but milder. It's probably just the window left cracked somewhere in the house, but it sits at the edge of awareness, easy to ignore, easy to follow into nothing. That's a little like what cerebrospinal fluid does. Sits just at the edge of your bodily awareness, close enough to be consequential, too quiet to ever notice directly. The question of what drives the fluid through the tissue, what generates the pressure to push it from the perivascular channels into the interstitial space and out the other side, is one that researchers are still refining. For a while, the leading idea was simple pressure gradient. Fluid enters under higher pressure from the arterial side and exits under lower pressure from the venous side. That made intuitive sense. Clean, mechanical. But the pressures involved don't always support it neatly. The math gets complicated. Some researchers have proposed that the movement is partly driven by osmotic forces, partly by the slow pulsations of arteries themselves, the vessel wall expanding with each heartbeat, squeezing the perivascular space, pushing fluid forward incrementally. Tiny pumps, everywhere, going all night. What the fluid picks up as it moves through tissue is, at this scale, invisible. Solutes, proteins, metabolic byproducts too small to see and too numerous to name individually. Amyloid beta, yes, but also tau protein, which is implicated in another set of neurodegenerative conditions. Also glutamate and potassium and other signaling molecules that accumulate in the interstitial space during neural activity and need clearing before the next day's activity can run cleanly. It isn't a targeted collection system. The fluid doesn't seek these things out. It just moves through and they dissolve into it, the way anything dissolves into moving water given time and contact. The biology here is more passive than it might seem, but passive isn't the same as careless. The exit route is still being mapped. What seems increasingly clear is that the fluid, once it has moved through brain tissue carrying its dissolved load, drains into lymphatic vessels along the dural sinuses, the large venous channels inside the thick outer membrane surrounding the brain. From there it enters the cervical lymph nodes in the neck, which means the waste from your brain ends up in the same lymphatic system that drains your tonsils and your throat. That always strikes me as odd. A little unglamorous, maybe. The brain's nightly cleaning, this quiet, careful, measured process, ends in the neck, in tissue that doesn't know anything about neurons. There's something I keep coming back to when I read about glymphatic research, which is how recently it was described. Not discovered exactly. The cerebrospinal fluid was known, the perivascular spaces were known, but understood as a functional clearance system. 2013, that's not long ago at all. And it makes me wonder, not anxiously, more with a kind of sleepy curiosity. What else is there operating this quietly that we haven't yet had the right tools to see? The body keeps things from itself in a way. Not secrets, just processes that are too slow or too small or too constant to register as events. The fluid has been moving through you every night of your life, and it took until 2013 for anyone to describe the system it belongs to with any precision. It doesn't mind. It was working either way. Right now the fluid is somewhere in the tissue, pressed between cells, dissolving what needs dissolving, following channels it didn't build through a brain that doesn't feel it happening. It will keep moving until morning. And the cells that hold those channels open, the ones whose feet press against the vessel walls, whose presence makes the pathway possible, they are doing something more than passive structural support. They're responsive, changeable. The channels they maintain don't simply exist. They're regulated. By what and how and why sleep changes that regulation, that's where the astrocytes come in. For most of the 20th century, If you'd asked a neuroscientist what the brain was made of, the answer would have centered almost entirely on neurons, which is understandable. Neurons are, by any measure, remarkable. Electrically excitable, structurally elaborate, capable of forming and reforming connections with a specificity that no human-engineered system has come close to replicating. They fire, they signal. They are, in the most direct sense, the stuff of thought. Everything else was considered scenery. The word glial comes from the Greek glia, meaning glue. It was coined in the 19th century by Rudolf Virchow, a German pathologist who, looking at brain tissue under the microscopes available to him at the time, saw something between and around the neurons, a kind of connective substance, not clearly cellular, not clearly doing anything, and named it for what it appeared to be: filler, structural adhesive, the tissue that held the important parts in place and had no particular importance of its own. Virchow was brilliant. He was also, on this point, almost completely wrong, though it would take the better part of a century for that wrongness to become undeniable. The cells he was looking at were not glue. They were, it turned out, doing things that neurons cannot do, managing functions that neurons depend on entirely, and operating with a quiet complexity that the tools of his era were simply not equipped to see. Not his fault. The limitation was technological, not intellectual. But the name stuck, and with it, for decades, a faint institutional dismissal. These cells are the matrix, the scaffolding, the supporting cast, and the neurons are the story. There is something almost uncomfortable about that history, if you sit with it. Generations of researchers built careers around neurons. The synapse, the action potential, the dendritic trees, the neurotransmitter, these became the grammar of neuroscience, the framework through which all questions about brain function were asked. And the glial cells were there the entire time, outnumbering neurons in some brain regions, consuming resources, expressing receptors, responding to signals, and largely being set aside as background noise. I don't say this critically. Science moves through the tools it has and the frameworks those tools suggest, but there's something worth pausing on in the fact that cells, which now appear to be central to the brain's nightly clearing process, spent most of modern neuroscience's history being described as passive. There are several types of glial cells. Oligodendrocytes wrap around axons in the white matter, producing the myelin that speeds electrical conduction. They have their own intricate biology, their own vulnerabilities, their own involvement in diseases like multiple sclerosis. Ependymal cells line the ventricles and are involved in cerebrospinal fluid production. Microglia are immune cells, derived from a completely different lineage than other brain cells, capable of movement and engulfment and a kind of cellular surveillance that has no real equivalent in the rest of the nervous system. And then there are astrocytes. Astrocytes are the most numerous glial cells in the human brain. Star-shaped, the name comes from the Greek for star, astron. They extend long, fine processes in every direction, and those processes make contact with blood vessels, with synapses, with other astrocytes, with the outer surface of the brain itself. A single astrocyte may contact tens of thousands of synapses. They tile the brain in a mosaic, each cell occupying its own territory, abutting but rarely overlapping with its neighbours, together covering essentially the entire brain volume. For a long time, even as neuroscience began to take glial cells more seriously, astrocytes were understood primarily as support. They maintain the chemical environment around synapses. They help regulate blood flow. They provide metabolic support to neurons. All of that is real, all of it matters, but the framing was still subordinate. They were doing important things in service of the neurons, which remained the primary characters. The smell of something warm, maybe a radiator running somewhere, maybe just the accumulated heat of a house settling for the night, drifts in and then isn't there. That's the quality of the astrocyte's presence in this story for a long time. Present, warm, functional, almost impossible to isolate from the environment it's part of. What changed the framing, at least partly, was the discovery of what astrocytes do at the blood-brain barrier and at the perivascular spaces. The channels, described now across 2 chapters, along which cerebrospinal fluid enters brain tissue. The end feet of astrocytes, the flattened terminal processes of those long branches, wrap around nearly the entire surface of blood vessels inside the brain. They form a secondary layer around the vessel wall, sitting just outside the endothelial cells that form the vessel itself. And embedded in those end feet, at extraordinarily high density, are water channels. Aquaporin 4, the specific channel involved, is a protein that allows water molecules to pass through cell membranes far faster than simple diffusion would permit. It is one of the most densely expressed membrane proteins in the entire body, and in the brain, it is concentrated almost entirely in the astrocyte end feet, precisely at the interface between the perivascular space and the broader tissue. This is not accidental. Those water channels are, in a significant sense, the mechanism by which cerebrospinal fluid moves from the perivascular channels into the brain tissue itself. The fluid arrives. It encounters astrocyte end feet. It moves through the aquaporin 4 channels, and in doing so it enters the tissue and begins its slow dissolving transit. The astrocytes are not passive walls, they are the gates, which makes the question of what regulates those gates, what opens them further, what narrows them, what changes their behaviour between waking and sleep, a question of considerable weight. Research in this area is still incomplete, which I think is worth saying plainly rather than glossing over with the summary that sounds more settled than it is. What appears to be true is that the state of astrocytes during sleep differs from their state during waking in ways that affect aquaporin 4 distribution and the permeability of the perivascular interface. Some work has suggested that astrocyte volume changes during sleep, that the cells themselves contract slightly, contributing to the expansion of the interstitial space described in the last chapter. Whether this contraction is driven by the astrocytes actively, or is a response to the changing ionic environment created by quieting neurons is not yet cleanly resolved. What is resolved? Astrocytes are not background. They are not yeasted glue. The name is a fossil, a record of an old misreading that has outlasted its usefulness. Rudolf Virchow looked at brain tissue in the 1850s and saw filler. He couldn't have seen otherwise. The staining techniques, the microscope resolution, the conceptual frameworks available to him, none of it would have made the astrocyte's function visible. He named what he saw honestly, and what he saw honestly looked like glue. What it was something more patient than that, something that had been doing its work long before anyone thought to look at it carefully, moving fluid through the brain's tissue each night, regulating the channels through which waste is cleared, tiling the entire neural volume in a quiet mosaic, unremarked, unnamed in any meaningful sense, operating without observation across every night of every human life for as long as there have been human brains. There's another kind of cell that works the same night shift. It doesn't stay still. You know the feeling by late afternoon. Not tiredness exactly, something heavier and less cooperative than tiredness. A kind of thickening behind the eyes, a reluctance that settles into the space between thoughts, and makes each one take slightly longer than it should. You reach for a word and it isn't quite there. You read a sentence and arrive at its end without having absorbed it. Something in the mechanism has slowed and you feel it, even if you'd never describe it in those terms. That feeling has a molecular address. It's been accumulating since the moment you woke up, not as mood, not as attitude, not as a story you're telling yourself about how tired you are, but as actual chemistry. Compounds building up in actual tissue. The sluggishness is not weakness, it's information. Every neuron that fires leaves something behind. This is not a metaphor. When a neuron generates an action potential, that brief, sharp reversal of electrical charge that constitutes a nerve signal, it requires energy, and burning that energy produces byproducts. Adenosine is one. It's a nucleoside, a component of ATP, the cell's primary energy currency, and it accumulates in the extracellular space as neurons burn through their fuel. It isn't toxic exactly, but it binds to receptors on neurons and other brain cells, and when enough of it is accumulated, the effect is unmistakable. A stubborn, heavy drag on alertness. A pressure towards stillness. You felt this pressure every day of your life. Adenosine is why. The coffee, if you had any this morning, worked by blocking adenosine receptors, not by reducing adenosine itself. The molecule kept accumulating, kept pressing its chemical case, and the caffeine simply stood in the way, occupying the receptor site so adenosine couldn't bind. Which is why, when the caffeine clears your system, the tiredness arrives with such blunt insistence. The adenosine was there the whole time, waiting. There's something almost stubborn about adenosine as a molecule. It doesn't rush. It doesn't spike dramatically at any particular moment. It just accumulates, incrementally, relentlessly, a slow chemical argument that the brain eventually cannot refuse. Sleep is, in part, the brain agreeing to that argument. During sleep, adenosine is cleared. The receptors open, the pressure lifts, and the glymphatic system moving its slow fluid through the interstitial spaces plays a role in that clearance, carrying adenosine and other metabolic waste out of the tissue, into the perivascular drainage, eventually out of the brain entirely. By morning, the slate is not blank, but it is meaningfully cleaner. Adenosine is the most discussed of the metabolic byproducts, partly because its link to sleep pressure is so well established, and partly because caffeine's mechanism of action made it a convenient research tool. You can study adenosine signaling by blocking it pharmacologically and watching what happens to sleep behavior. But it isn't alone. Glutamate accumulates in synaptic clefts during prolonged neural activity. Potassium ions spill into the extracellular space with every action potential, and the concentrations of both shift the electrochemical environment in ways that, if uncorrected, would eventually impair the ability of neurons to fire at all. The brain's ionic balance, the precise ratios of sodium, potassium, calcium, and chloride inside and outside cells, is not self-maintaining. It requires active correction. It requires the kind of slow, deliberate restoration that waking activity doesn't fully allow. Somewhere in the tissue right now, ion pumps are running. They run continuously, but they run better, more completely with less interference, when the neurons they're serving have gone quiet. Outside, something has shifted in the night air. A truck somewhere far off moving through a neighborhood that isn't this one. The sound reaches as a low, soft growl and then doesn't. The room reasserts its quiet. This is what the brain is doing in its own way, reasserting a quiet that activity had disturbed. Not silence exactly, more like the return to a baseline that waking life pulls you away from and sleep patiently reinstates. There is a concept in neuroscience called synaptic homeostasis, and it carries more weight than its clinical name suggests. The hypothesis, developed most extensively by Giulio Tononi and Chiara Corelli at the University of Wisconsin, proposes that waking activity, learning, perceiving, moving through the world, systematically strengthens synaptic connections throughout the brain. Each experience, each moment of attention, each new piece of information encountered and encoded adds a small increment of synaptic weight somewhere. The connections between neurons become, on average, slightly more potent with each passing hour of wakefulness. This sounds like a good thing. And in small doses it is. Synaptic strengthening is how learning works, how memories form, how the brain adapts its structure to reflect experience. But there is a cost. Stronger synapses require more energy to maintain. A brain in which every connection is maximally potentiated would be metabolically unsustainable. And it would also, paradoxically, be worse at learning, because the signal-to-noise ratio that allows new information to register above baseline would be compromised. You cannot write on a page that is already completely covered. Sleep, the hypothesis proposes, allows a global downscaling of synaptic strength. Not erasure, not the loss of memories or skills. A proportional reduction, all connections scaled back together, so that the relative differences between them, which encode what was learned, are preserved. The brain doesn't lose the content, it recovers the capacity. This hypothesis is still actively debated. I want to be clear about that because synaptic homeostasis is sometimes described as settled fact, and it isn't. It's a well-supported framework with compelling experimental evidence, and also real critics who point to findings that don't sit neatly within it. Some studies have found that certain synapses strengthen during sleep rather than weakening, which complicates the clean version of the story. The biology is, as biology tends to be, more tangled than any single elegant hypothesis fully captures. What isn't debated? Something happens to synapses during sleep that doesn't happen during waking. The fine structure of synaptic connections changes between going to sleep and waking up. The mechanisms involved are multiple and not yet fully catalogued, and the result, the refreshed, recalibrated feeling of a brain that has slept well, is something every person reading this has experienced, even if the cellular explanation for it remains partially unresolved. The neurons are not the only ones doing the leaving behind. As they fire and communicate and burn through their fuel, the molecular environment surrounding them shifts in ways that other cells must respond to. Astrocytes, which tile the brain in their quiet mosaic, are exquisitely sensitive to the extracellular environment. They take up excess glutamate from synaptic clefts, helping to terminate signaling and prevent the kind of sustained excitation that becomes damaging. They redistribute potassium, taking up the excess that spills out with each action potential and moving it away from sites of high activity. They are, in this sense, cleaning up as the neurons go. And they do this continuously, not just at night, but throughout the day, in real time, as neural activity generates its constant small debris. Sleep doesn't initiate this function. Sleep makes it more complete. The difference between the two is the difference between mopping a floor while people are still walking on it and mopping it after the room has emptied. Both matter. One finishes the job. What your neurons leave behind is by itself not dangerous. The amounts are small. The systems that clear and correct are robust. A single night of poor sleep doesn't cause lasting damage to a healthy brain. The research is clear on that, and it's worth saying explicitly, without softening it into something more alarming than it is. The concern emerges from patterns, from chronic sleep restriction, from the repeated, accumulated failure to clear what accumulates, amyloid beta building toward plaque, synaptic weights never fully rescaled, ionic gradients never fully restored. A single night. That's all tonight is, and tonight the clearing is running. The adenosine is lifting. Somewhere in the tissue, glutamate concentrations are dropping back toward the baseline. Potassium is being redistributed. The synaptic machinery that spent the day building and strengthening is, in some regions and by some mechanisms, beginning the slow process of recalibration. You don't feel any of this. There's nothing to feel. It happens below sensation, in the gaps between cells, in the quiet chemistry of a brain that has, for now, stopped performing and started repairing. The cells doing the most visible part of that repair, the ones moving through the tissue rather than staying fixed within it, haven't been introduced yet. They come next, and unlike the astrocytes, they are not content to stay in one place. Start with something you already know without knowing you know it. Your body is not solid, not in the way it feels, not in the way it presents itself when you press a hand against a surface or feel the weight of your own head against a pillow. It seems solid, dense, continuous. But at the scale where biology actually operates, the body is mostly space. Gaps between molecules, gaps between cells, gaps between structures that look at the resolution of ordinary experience, like they are pressed seamlessly together. The brain is no different. Between every neuron and its neighbours, between the astrocytes and the blood vessels, and the myelin-wrapped axons running their long distances through white matter, there is space. Real, fluid-filled, measurable space. It's called the interstitial space, or sometimes the extracellular space, and in the brain it accounts for somewhere between 15 and 20% of the total tissue volume in a waking adult. 15 to 20% sounds modest. It isn't really. Scaled to the volume of the human brain, roughly 1,200 to 1,400 cubic centimeters depending on the individual, that's somewhere between 180 and 280 milliliters of space that is not cell, not vessel, not membrane, just gap, threaded through everything, continuous and interconnected, holding the extracellular fluid in which neurons and glia are, in a very literal sense, suspended. You're not quite as solid as you feel. What happens to that space during sleep is one of the more quietly startling findings to come out of glymphatic research. It expands, not dramatically. This isn't a sudden restructuring, nothing shifts or lurches, but measurably, consistently, and in ways that appear to be a precondition for the glymphatic clearance described across the earlier chapters. The interstitial space in sleeping brain tissue is estimated to be roughly 60% larger than in waking tissue. The same cells, the same architecture, the same basic structure, but the gaps between everything widen, and the fluid that has been pressing at the edges of those gaps presses further in. 60% is not a small number. The question of how this expansion happens is, and I want to be direct about this, not fully answered. What appears to be involved is the behavior of neurons themselves when they quiet. Active neurons accumulate potassium in the extracellular space with every action potential they fire, and high extracellular potassium causes cells to swell slightly, drawing water in across their membranes through osmotic pressure. When neurons go quiet, when the barrage of firing that characterizes waking brain activity settles into the slower, more synchronized rhythms of sleep, the ionic environment changes. Potassium concentrations in the extracellular space drop. Cells release some of the water they had drawn in, and the spaces between them open. There's a particular quality to this that I find difficult to describe without reaching for something spatial. The feeling of a room after the people have left it. Not empty. Still furnished, still warm, still holding the shape of everything that was happening an hour ago, but with a looseness to the air that wasn't there before, a capacity for movement that the crowd had displaced. The brain's interstitial space in sleep has something of that quality, not empty, not inert, but open in a way that it wasn't. And into that openness fluid moves. The glymphatic system doesn't create the space, it uses it. This distinction matters more than it might seem. The channels along which cerebrospinal fluid enters brain tissue, the perivascular spaces lined with astrocyte end feet, deliver fluid to the edge of the interstitial compartment. From there, the fluid moves through the tissue itself, driven by pressure gradients and possibly by osmotic forces and the slow pulse of arterial walls. But the distance it can travel, the volume of tissue it can reach, depends on how much space the tissue offers. Waking tissue offers less. Sleeping tissue offers more. The system and the space are, in this sense, partners, neither sufficient without the other. The fluid has the channels and the pressure and the aquaporin four gates, but without the expanded interstitial space that sleep creates, it cannot reach deep enough to matter. A light is on somewhere in a room down the hall, the thin line of it visible under a door, not bright enough to be a problem, just present. The house does that sometimes, holds small lights in its corners, and the darkness around them is fuller for it. The interstitial space is a little like that gap under the door, thin, easy to overlook, doing something quietly useful that you'd only notice if it weren't there. There are 4 things the interstitial space does that don't get described often enough together. It provides the medium through which signaling molecules diffuse between neurons that aren't synaptically connected. It holds the ionic environment that determines whether and how neurons can fire. It serves as the pathway for glymphatic clearance, and it buffers the mechanical forces that reach the brain through blood pressure and movement. 4 things, none of them small, and all 4 are affected by how much space there is, by whether the tissue is in its waking, compressed configuration or its sleeping, expanded one. The term for signaling that happens through diffusion across the interstitial space, rather than across a synapse, is volume transmission. It's a slower kind of communication, less targeted, more ambient. Neuromodulators like dopamine and serotonin and norepinephrine are released in some regions and diffuse outward, reaching receptors across a wider area than a synapses would allow. The interstitial space is the medium they diffuse through. During sleep, when the space is wider, the dynamics of that diffusion change. How the broader space affects the concentration gradients, the distances reached, the receptors engaged, is an area of ongoing research. What seems clear is that the geometry of the interstitial space is not a passive background. It participates. It shapes the signaling that happens within it. Something about the concept of the interstitial space tends to stay with me longer than other concepts in this area. Maybe it's the inversion it requires. You spend a life thinking of the brain as substance, as a dense, continuous, bounded thing. And it is those things. But it is also simultaneously a structured arrangement of gaps. The function lives in both. The cells and the spaces between them are equally part of the architecture, equally part of what makes the whole thing work. Neither solid nor empty, both held together. By morning, when wakefulness begins to return, when neurons start firing in their waking patterns and potassium begins to accumulate again in the extracellular space, the interstitial volume will quietly compress back toward its daytime configuration. The gaps will narrow. The fluid that moved through them in the night will have done its work and drained. The space that made the clearance possible will recede, and the brain will return to the denser, more compressed state it keeps during the hours it spends attending to the world. It will do this again tomorrow night, and the night after. The space has been opening and closing this way, on this same quiet schedule, since long before there was a word for it or a tool capable of measuring it. Unannounced, unremarkable to anything except the fluid that needs it. The opening, the moving through, the closing again, What moves through that space and what happens to the signals it carries and the waste it collects depends on more than fluid and architecture. It depends on cells that are watching, sampling, moving through the tissue the way the fluid does, but with intention, not passive, not fixed. Those cells have been waiting for several chapters now. They are ready. You have felt this before, not as a concept, as a physical fact, as something that happens in the body with the same reliability as hunger or cold, something that arrives on its own schedule regardless of what you'd prefer. By mid-afternoon, or sometimes earlier if the day has been particularly relentless, there is a weight that settles somewhere behind the eyes and doesn't shift. Not pain, not quite discomfort, a kind of thickening, a reluctance that spreads from thinking into movement into the simple act of holding attention on anything at all. You know this weight. You've probably tried to push through it. Most people do. Another cup of something, a change of room, a walk to the window and back, and small negotiations with the process that doesn't negotiate. The weight lifts temporarily, or seems to, and then returns, a little heavier than before. That weight has a name, and the name, until tonight, probably meant nothing to you. Adenosine is a small molecule. Chemically, it belongs to a class called nucleosides, a nitrogenous base attached to a sugar, compact and structurally plain, the kind of molecule that appears in biochemistry diagrams without drawing much attention to itself. It is a component of adenosine triphosphate, ATP, which is the primary energy currency of nearly every cell in the body. When ATP is broken down to release energy, when a neuron fires and burns through its fuel to generate the electrical charge that makes signaling possible, adenosine is among the residue that remains. It accumulates. This is not a flaw in the design, or at least it doesn't appear to be. Adenosine accumulation in the extracellular space around neurons seems to function as a kind of running chemical record of how long and how hard those neurons have been working. The more activity, the more adenosine. The more adenosine, the stronger the signal toward rest. It is, in its quiet way, one of the more elegant biological feedback mechanisms in the brain, the very fuel of wakefulness, leaving behind the compound that makes wakefulness unsustainable. The work contains its own ending. Adenosine binds to receptors on neurons and on glial cells, and the binding has inhibitory effects. It quiets excitability, slows signaling, reduces the brain's overall inclination toward the kind of rapid, sustained activity that waking life demands. There are several adenosine receptor subtypes, but the one most involved in sleep pressure is called A1, widely distributed across the brain, particularly concentrated in regions involved in arousal and attention, and exquisitely sensitive to accumulating adenosine in the way that a scale is sensitive to accumulating weight. The pressure builds across the day not in a straight line, but in something more uneven than that. Faster during periods of intense cognitive work, slower during rest, modified by stress hormones and light exposure, and the half-dozen other signals the brain uses to locate itself in time. But the direction is consistent. Adenosine goes up. The pressure towards sleep goes up with it. This is what sleep researchers call sleep pressure, or sometimes sleep drive, the accumulated homeostatic need for sleep that builds with every hour of wakefulness and is relieved specifically and almost exclusively by sleep itself. Caffeine works by impersonation. The adenosine receptor, specifically A1 and also A2A, has a binding site that caffeine fits into with uncomfortable precision. Not because the receptor evolved to accommodate caffeine, but because caffeine's molecular structure happens to resemble adenosine closely enough to occupy the same space. Caffeine binds, adenosine cannot. The inhibitory signal that adenosine would have sent goes unsent, but the adenosine is still there. This is the part that tends to surprise people when they encounter it clearly for the first time. Caffeine does not reduce adenosine levels. It does not slow adenosine production. It does not interfere with adenosine's accumulation in the extracellular space. It simply stands in front of the door. The molecule knocks repeatedly and finds the entrance occupied, and while it waits, it accumulates further, which is why the crash, when caffeine clears the receptors, has that particular blunt quality, less like tiredness returning and more like tiredness arriving all at once, carrying everything it had been holding. The afternoon wait, deferred and then delivered. Something outside, a branch against a window maybe, or just the house making one of those low, unidentifiable sounds that houses make in the cold, settles into silence, then nothing. The night re-establishes itself. During sleep, adenosine is cleared. The mechanisms involved are multiple and still not exhaustively mapped. But what is established is that glymphatic fluid movement plays a meaningful role. Adenosine, dissolved in the interstitial fluid that the glymphatic system flushes through brain tissue, is carried outward along with other metabolic residue. Additionally, adenosine is taken up and metabolized by astrocytes, which express the enzymes capable of breaking it down. The molecule that accumulated across the day is, across the night, gradually removed. By morning, in a brain that has slept adequately, adenosine concentrations are near their daily low point. The receptors that were occupied or waiting to be occupied are open. The sleep pressure that had been building since the previous morning is, for now, discharged. This is why morning feels different from afternoon. Not just subjectively, chemically. The brain that wakes after sufficient sleep is operating in a genuinely different molecular environment than the one that was struggling through hour 14 of wakefulness. The difference isn't mood, it's chemistry that mood follows from. There is something I've always found quietly affecting about adenosine as a concept, the way it makes the need for sleep into something tangible, something that accumulates in real tissue at measurable concentrations rather than existing as a vague behavioral preference. People sometimes talk about sleep as a luxury, or frame the need for it as a weakness, as if the urge to lie down and stop were a failure of discipline rather than a biological process as fundamental as hunger. Adenosine makes that framing difficult to maintain. The weight you feel is real. It is molecular. It is the residue of the brain's own work, building up in the spaces between cells, binding to receptors, with a chemical insistence that no amount of willpower can permanently override. You cannot decide not to need sleep any more than you can decide not to need glucose. The body has already decided. Right now, adenosine concentrations in your brain are lower than they were this afternoon. The clearance has been running since you closed your eyes. The glymphatic fluid pressing through the expanded interstitial space. The astrocytes taking up what they can metabolize. The slow chemistry of restoration doing what waking never fully allows. The weight is lifting, not gone yet. Still lifting. Somewhere in the tissue, the receptors that adenosine occupied through the long hours of the day are releasing their load. The inhibition is becoming unnecessary. The signal that your brain has been sending toward rest, that stubborn, heavy drag that arrived every afternoon and was ignored or deferred, is being answered. You are answering it now, the other molecules that sleep clears. The proteins, the ionic imbalances, the synaptic residue described in the last chapter, clear on their own timescales through their own mechanisms. But adenosine, in a way, is the one that you carry into every night as a felt experience rather than a chemical abstraction. The one that arrives as sensation before it arrives as biology. The weight behind the eyes, the slowing of thought. The moment, sometime in the late evening, when holding your eyes open begins to feel like a task rather than a default. That was adenosine, and now, in the dark, it is leaving. Sleep doesn't arrive. That's not quite how it works, even though it feels that way, like something external coming toward you, like a tide you wait for. It's more that wakefulness gradually loses its argument. The systems that maintain alertness, that keep the brain's activity above a certain threshold of coherence and responsiveness, begin to lose ground. Not all at once, not in a single moment that could be identified and marked. Piece by piece, region by region. The transition from wakefulness to sleep is, at the cellular level, a negotiation between competing systems, and the word negotiation is not quite right, but it's closer than the alternatives. There are systems in the brain whose activity promotes wakefulness. The locus coeruleus releasing norepinephrine, the raphe nuclei releasing serotonin, the tuberomammillary nucleus releasing histamine, the lateral hypothalamus releasing orexin. These are not independent actors. They form a loosely coordinated network of arousal, each reinforcing the others, each keeping the overall system above the threshold where coherent waking activity is possible. And opposing them, or rather accumulating against them, is the homeostatic pressure described in the last chapter. Adenosine, sleep pressure. The chemical weight of a day's worth of work pressing on the receptors that the arousal systems depend on. The balance shifts, slowly then less slowly. Orexin is worth a moment. It's a neuropeptide, a small protein used for signaling, produced by a surprisingly small cluster of neurons in the lateral hypothalamus, a region toward the sides and middle of the brain. The neurons that produce orexin number somewhere in the tens of thousands in a human brain, which is a vanishingly small population relative to the roughly 86 billion neurons total. And yet the orexin system appears to function as a kind of stabilizer for the entire wake-sleep transition, not driving wakefulness so much as preventing the brain from sliding unintentionally between states. The evidence for this comes starkly from what happens when the orexin system fails. Narcolepsy, the condition characterized by sudden, uncontrolled transitions into sleep or sleep-like states, sometimes accompanied by cataplexy, a sudden loss of muscle tone triggered by strong emotion, is in most cases caused by the loss of orexin-producing neurons. The immune system, for reasons that remain incompletely understood, destroys them, and without those neurons, the transition mechanism loses its governor. Wakefulness becomes unstable. Sleep intrudes without warning. The boundary between states, it turns out, requires active maintenance. There is something. I keep returning to this, maybe because it seems important and maybe because it just interests me more than it should. There is something almost precarious about the wake-sleep boundary when you look at it this way. Not dangerous, but fragile in a structural sense. The brain doesn't sit solidly in one state and then solidly in another. It maintains each state against the competing pull of the other. The arousal systems hold wakefulness together. The homeostatic pressure dismantles it incrementally across the day. And somewhere in the evening, for most people, the dismantling wins. A threshold is crossed. What happens at the level of individual neurons during this crossing is not a simple quieting. Some neurons slow their firing, some change the pattern of their firing, moving from the irregular, tonically active patterns characteristic of wakefulness to more synchronized rhythmic patterns as the brain enters non-REM sleep. The synchronization is important. It's not silence. It's a different kind of activity, organized differently, serving different purposes, less responsive to sensory input from the outside world, and more directed toward the internal processes of consolidation and clearance. The electroencephalogram, the EEG, the recording of electrical activity across the scalp, shows this transition clearly. Waking brain activity is characterized by fast, low-amplitude waves, the electrical signature of many different neural populations doing many different things, their individual contributions blending into something that looks, at the scale of an EEG electrode, like relatively rapid, relatively small fluctuations. As sleep approaches, the waves slow. Their amplitude increases. The brain is not doing less. It is doing something more coordinated, more synchronized, more like a single thing and less like a crowd. The thalamus plays a central role in this. It sits near the center of the brain, a paired structure of grey matter that serves in waking as the primary relay for sensory information on its way to the cortex. Visual input, auditory input, touch, proprioception, most of it passes through the thalamus, which processes and forwards it, shapes what reaches conscious awareness and what doesn't. The thalamus is, in waking, partly responsible for the brain's connectivity with the world outside it. During sleep onset, the thalamus does something unexpected. It begins generating its own rhythmic activity, sleep spindles in the EEG terminology, bursts of oscillatory activity at around 12 to 15 cycles per second, generated by circuits within the thalamus itself and projected outward to the cortex. These spindles appear to serve a gating function. They block the relay of sensory information to the cortex, reducing the brain's responsiveness to external stimuli, helping to maintain sleep against the disruptions that the outside world continuously offers. The thalamus, essentially, closes the door. The carpet in this room, or whatever is underfoot, whatever texture the floor holds tonight, has a smell that only appears at night for some reason. Or maybe it's always there and daytime is too loud to carry it. Something dusty and faintly warm. It's easy to ignore. Almost everything is when the thalamus starts doing its work. The channels mentioned in this chapter's title are not a single structure. They are, more accurately, the sum of a transition, the collective opening of sleep, the thalamic gates closing to sensory input while opening to internal oscillation, the ion channels in neuronal membranes shifting their behavior as the membrane potential drops toward the patterns of slow-wave sleep, the aquaporin 4 channels in astrocyte end feet allowing more fluid through as the interstitial space expands, the adenosine receptors releasing their load as clearance accelerates. Many channels, many thresholds, all of them crossed, or crossing, right now. There is a stage before deep sleep that is worth noting, not as a prelude or exactly, but because it has its own character. Stage 1, non-REM. Sleep is brief, a few minutes typically. The brain is still close to the surface, still capable of being pulled back by a sound or a shift in the room. Muscle tone decreases. The eyes move slowly under closed lids, not in the rapid way of REM, but in a slow rolling drift. Hypnic jerks sometimes occur here, the sudden involuntary muscle contractions that many people experience as a falling sensation, startling them back to wakefulness. The mechanism behind hypnic jerks is still debated, though one proposal is that the motor system, losing the suppressive signals that waking muscle tone depends on, fires briefly and uncoordinatedly as it adjusts. The body briefly not sure where it is. Stage 2 arrives. Sleep spindles appear on the EEG. K-complexes, large sharp waveforms that arise in response to external stimuli and seem to serve a protective function, suppressing arousal, become visible. The brain is deeper now, more committed. The sensory gate that the thalamus has been closing is mostly closed. And then, in time, after the spindles and the K-complexes and the slow drift through stage 2, the slow waves arrive, delta waves, large, slow, sweeping, the signature of deep non-REM sleep, of slow wave sleep, which is where most of what has been described across these chapters, the glymphatic surge, the adenosine clearance, the synaptic downscaling, happens with the most intensity, where the channels in every sense are most fully open. You're somewhere in this now, not at the beginning of the transition anymore. Deeper. The arousal systems have lost enough ground that the boundary feels less precarious than it did an hour ago. The thalamus is doing its quiet gatekeeping. The slow ways, if they haven't arrived yet, are not far. The negotiation is mostly over. Wakefulness didn't lose exactly, it just stopped insisting. What moves through the open channels while you're here? What arrives in the tissue? What gets carried out? What happens to the cells that have been waiting for precisely this configuration of quiet is where the next part of the story lives. And the cells that have been waiting the longest, the ones that spent the waking hours extending and retracting, sampling and assessing, moving through the tissue with a patience that has no human equivalent, they are already moving. They move. That is where this has to start, because it changes everything else about them. Most cells in the body are, in the relevant sense, stationary. They occupy a position, perform a function, divide when required, die when required, and do not travel. Neurons extend long processes across considerable distances, but the cell body stays put. Astrocytes tile their territory and remain within it. Even the blood moving through the brain's vessels is carried. It does not move itself. Microglia move themselves, not quickly, not in any way that would be visible to ordinary perception. This is cellular motion, measured in micrometers per hour, the kind of displacement that only becomes apparent when time-lapse imaging compresses hours into seconds. But the movement is real, and it is continuous, and it is purposeful in a way that sets microglia apart from nearly everything else in the central nervous system. They extend processes, thin, branching projections that reach outward from the cell body in multiple directions simultaneously, and then retract them, and then extend again in a ceaseless cycle of exploration. A single microglial cell can survey its entire local territory. The small volume of brain tissue it considers its domain in a matter of hours. It is sampling the environment, tasting the chemical composition of the extracellular space, assessing the surfaces of nearby cells, checking for signals that indicate damage or infection, or the kind of molecular disorder that requires a response. Most of the time it finds nothing requiring action, and it keeps looking anyway. The word microglia means approximately small glue, the same glial root as astrocyte, the same historical dismissal embedded in the name. But microglia are not small versions of the other glial cells. They are not even, in the strict developmental sense, brain cells at all. They come from somewhere else. During early embryonic development, before the brain has taken its final form, a population of primitive immune cells called macrophages migrates from the yolk sac into the developing central nervous system. This happens early, before the blood-brain barrier closes, before the neural tube has fully differentiated into what will become the mature brain and spinal cord. Once inside, these cells take up permanent residence. The blood-brain barrier closes around them, and unlike nearly every other cell in the adult brain, they are not replaced by cells derived from neural stem cells. They maintain themselves through local self-renewal, a population apart, with a lineage that traces back not to the neuroectoderm, but to primitive myeloid progenitors in the embryonic yolk sac. Every microglial cell in your brain right now is descended from cells that migrated into the developing nervous system before you were born. They have been there in some continuous cellular lineage your entire life. For most of the 20th century, and here the history rhymes uncomfortably with what was said earlier about astrocytes, microglia were understood primarily as the brain's immune sentinels, present for pathology, relevant in disease. When something went wrong, infection, injury, neurodegeneration, microglia activated, changed shape, proliferated, engulfed debris and pathogens and damaged cells. They were the brain's emergency response. In health, in the normal brain, it was assumed they were relatively quiet, monitoring perhaps but not doing much, resting. The word used for their normal state was, for decades, resting microglia. It was not a good description. What imaging technology eventually revealed, particularly two-photon microscopy, which allowed researchers to watch individual cells in living brain tissue in real time, was that microglia in a healthy brain are not resting. Not remotely. The constant process extension, the continuous territorial surveillance described above, is not the behavior of a cell in standby. It is the behavior of a cell with an ongoing job that happens not to involve emergency response. The question became, what is that job? And the answer, still being assembled, is more various and more consequential than anyone expected when the question was first properly asked. One of the functions now attributed to microglia in the healthy brain is synaptic pruning. The term is borrowed from horticulture, the deliberate removal of growth to improve the overall structure, and it describes something that microglia appear to do throughout life, though with particular intensity during early brain development. Synapses, the junctions between neurons across which signals are transmitted, are formed in enormous excess during development. The developing brain overbuilds its connectivity, generating far more synaptic connections than the mature brain will ultimately maintain, and then selectively eliminates the weaker or less active ones, a process called synaptic pruning, which continues at a lower rate into adulthood and appears to occur with renewed intensity during sleep. Microglia participate in this by engulfing synaptic material. They extend a process toward a synapse, make contact and consume part of it. The material is taken into the cell body and broken down. The synapse or the specific synaptic element that was engulfed is gone. This sounds destructive. It is, in the most precise sense, the opposite. There is an old radio somewhere in the building, not playing, just sitting on a shelf in a room that probably doesn't get used much, his dial set to a station that may not exist anymore. A detail that has no reason to be here except that it is the kind of thing the mind reaches for at this hour, when the border between what's real and what's assembled from memory gets soft. The selectivity of microglial pruning is one of the more actively studied questions in cellular neuroscience. How does a microglial cell know which synapse to engulf and which to leave? The answer appears to involve molecular tags. Complement proteins, primarily C1q and C3, which are components of the immune system's complement cascade and are deposited on synapses that are relatively inactive or weakened. Microglia express complement receptors. They can read the tags. A synapse bearing a complement marker is a synapse flagged for removal. The biology is more complicated than this summary suggests. Complement tagging is not the only signal involved, and the conditions under which synapses are tagged versus protected are still being worked out. But the basic architecture is, neurons signal their own weakness through complement deposition, and microglia respond to that signal by pruning. A kind of managed demolition, continuous and selective, operating just below the threshold of anything that could be felt. During sleep, microglial pruning activity appears to increase. This is consistent with what was described in the synaptic homeostasis hypothesis from an earlier chapter. The idea that sleep allows a global downscaling of synaptic strength, a recalibration of the connectivity that waking experience has incrementally potentiated. If sleep is when the brain reduces synaptic weight across the network, then sleep is also when the physical removal of synaptic material, which microglia perform, would be most active. The two processes are related. They may be, in some respects, the same process viewed at different scales, but pruning is not the only thing microglia do during sleep. There is evidence, more recent, more preliminary, and appropriately caveated, that microglial process motility itself changes across the sleep-wake cycle. During wakefulness, processes extend and retract at a relatively steady rate. During sleep, some studies have found an increase in the reach and speed of process extension, microglia surveying their territory more aggressively, covering more ground, spending more time in contact with synaptic structures. Why this might be? What drives the change in motility? What signals tell a microglial cell that the brain has entered a state where more intensive surveillance is appropriate is not yet cleanly understood. Norepinephrine may be involved. The locus coeruleus, which releases norepinephrine throughout the brain during waking, has been shown to suppress microglial process motility. High norepinephrine, less movement. During sleep, locus coeruleus activity drops, norepinephrine levels in the brain fall, and microglia appear to extend their processes more freely. The arousal system that keeps you awake is, in this sense, also the system that keeps microglia somewhat in check. Sleep releases the restraint. There is something about the image of microglia moving more freely through sleeping brain tissue that I find difficult to let go of. Not because it is dramatic, but because it is so completely invisible from the outside. You lie still, your breathing slows. From any external vantage point, very little appears to be happening. And inside, in the tissue that produces everything you are, thousands upon thousands of small cells are extending and retracting, reaching towards synapses, reading molecular tags, engulfing what needs engulfing, releasing what needs releasing, surveying a territory that they have claimed and maintained since before you were capable of any experience at all, not resting, working. Microglia also release signaling molecules, cytokines among others, that influence sleep itself. Interleukin-1 beta and tumor necrosis factor alpha, both produced by microglia, have been shown to promote slow-wave sleep when administered experimentally. The relationship appears to be bidirectional. Sleep modulates microglial behavior, and microglia, through their signaling, modulate sleep. The cells are not simply responding to the sleep state, they are in some degree participating in its regulation. A cell that helps create the conditions for the process that allows it to do its most important work. The other thing worth saying about microglia, and this feels important enough to say plainly rather than weaving it into something more elaborately constructed, is that their dysfunction is implicated in a growing list of neurological and psychiatric conditions. Excessive synaptic pruning during early development has been linked to schizophrenia. Insufficient pruning, or pruning that fails to target the right synapses, has been proposed as a factor in autism spectrum conditions. Microglial dysfunction appears in the pathology of Alzheimer's disease, in the chronic neuroinflammation that follows traumatic brain injury, in the neural changes associated with depression. These are not simple relationships. The research is complex, contested in places, and far from producing clinical applications that match the ambition of the underlying findings. But the direction of the evidence is clear enough to take seriously. Getting this right, the pruning, the surveillance, the cytokine signaling, the sleep-dependent motility, matters for the brain's long-term integrity in ways that extend well beyond any single night. Tonight, in sleeping tissue, a microglial cell somewhere in your cortex is extending a process toward a synapse that fired weakly and infrequently throughout yesterday. It makes contact, it reads whatever molecular signal the synapse has been tagged with. It considers, and this is not the right word, but there is no better one at this scale. It considers. The process withdraws, or it doesn't. Either way, the cell body moves on, retracting and extending again in a different direction, into a different small volume of tissue, sampling a different molecular neighborhood with the same patient. Unhurried thoroughness that it has brought to every night of your life. It does not know it is being described. It does not need to. Not every connection your brain made today should survive the night. This isn't a harsh claim. It isn't even really a claim about failure. It's closer to something structural, an observation about how the brain manages its own complexity over time, how it keeps itself from being buried under the accumulated weight of every momentary response it has ever generated. The brain builds too much, on purpose. During development, this overbuilding is dramatic and deliberate. The infant brain generates synaptic connections at a rate that will never be matched again, forming in the first years of life far more connections than the mature brain will ultimately keep. The excess is not a mistake. It appears to be a strategy. Build widely first, then refine. Let experience determine what stays. What shapes the refinement is activity. Synapses that are used, that participate in repeated coordinated firing that carry signals reliably across the junction between neurons, are preferentially stabilized. Synapses that are weak, redundant, rarely activated, or simply outcompeted by stronger neighboring connections are candidates for removal. The process has a name taken from gardening, pruning. The metaphor works well enough to be useful and badly enough to be worth setting aside fairly quickly. A gardener prunes intentionally with a plan, shaping growth towards some imagined future form. The brain's pruning has no gardener. It is driven by molecular signals and cellular responses and the statistics of neural activity, a process that produces refinement without anyone, at any level of the system, intending refinement. It just emerges from the rules. The cellular agent of pruning, described in the last chapter, is the microglial cell. But the mechanism by which microglia identify which synapses to engulf and which to leave involves proteins from a system that predates the brain by a considerable evolutionary margin. The complement system is part of the innate immune response, one of the oldest components of vertebrate immunity, present in fish, in amphibians, in every lineage that has had a recognizable immune system for the last several 100 million years. It operates through a cascade of proteins that tag pathogens and damaged cells for destruction, marking them with molecular flags that other immune cells can recognize and respond to. In the brain, some of those same proteins, C1q, C3, and their downstream effectors, are deposited on synapses, specifically on synapses that are relatively weak or inactive. The microglia express receptors for C3. They find the tagged synapses. They engulf them. The immune system's oldest tagging mechanism, repurposed for the brain's own internal editing. Something about that repurposing is worth sitting with. The complement system evolved to mark things that didn't belong: foreign cells, damaged tissue, pathogens. And somewhere in evolutionary time, the brain recruited it for a different task entirely, marking its own connections for removal. Not because they are foreign, not because they are damaged, simply because they are not being used enough to justify the metabolic cost of maintaining them. The machinery of immune recognition became the machinery of neural refinement. A window somewhere is doing that thing where it isn't quite closed. Not open enough to feel, just enough to carry a faint sound of outside air pressing against the frame. The kind of thing you only notice once and then your attention releases it. In adults, synaptic pruning continues at a lower rate than in early development, but it does not stop. And during sleep, particularly during slow-wave sleep, when microglial process motility increases and the norepinephrine suppression of microglial activity is lifted, the rate of pruning appears to increase relative to waking hours. This fits with what the synaptic homeostasis hypothesis predicts, described 2 chapters back. If waking experience incrementally strengthens synaptic connections across the brain, and if sleep restores the overall synaptic weight toward a sustainable baseline, then sleep must involve not only molecular downscaling but physical removal of synaptic material. The removal is real. It is structural. By morning, some dendritic spines, the small protrusions on the receiving side of synapses, will be gone, not weakened, not modified. Gone. Dendritic spines are strange structures to think about. They are small, on the order of 1 to 2 micrometers in length, with a head diameter often under a micrometer. A single neuron may carry thousands of them, studying its dendrites like nothing actually. There is no clean comparison that doesn't flatten something important. They are just what they are. Tiny postsynaptic specializations, each one the receiving end of a synaptic connection, each one representing a relationship between 2 neurons that took energy to build and takes energy to maintain. Some of them will not be there tomorrow. The selectivity of the process is what prevents it from being destructive. A synapse that has been used, that fired repeatedly and reliably enough to have been potentiated, to have accumulated the molecular machinery of an active connection, carries different molecular markers than one that has been quiet. The tagging and the reading of tags is sensitive to activity history in a way that means the pruning is not random and not indiscriminate. What was important survives. What was provisional does not. This is why the popular description of sleep as the time when memories consolidate is true but incomplete. Sleep consolidates, yes. It stabilizes and integrates the experiences of the day into longer-term neural structure. But it also removes. And the removing is not the opposite of the consolidating. The 2 processes work together. You cannot write more clearly on a page by only adding ink. Some things are kept by letting other things go. What remains in the morning is not less than what was there at night. It is more exact. The network that wakes with you has been edited, not by anything that had a plan, not toward any particular goal, but by the accumulated logic of activity and disuse and molecular signaling working at the scale of individual synaptic junctions across billions of cells. The feeling of a rested brain is not only the feeling of adenosine cleared and lymphatic waste removed. It is also, quietly, the feeling of a network that has released what it no longer needed. There is something in that which, the tissue is quieter now than it was an hour ago, not silent, just reduced, in the right places, by the right amount, in a way that will not be noticed until tomorrow, when a thought arrives more cleanly than expected, or a connection forms more easily, or something that was tangled during the long afternoon has, without any intervention, come loose. The pruning will continue for a while yet. There is more to release. Inside every cell all the time, proteins are being made. This is not a remarkable statement. It is simply what cells do, as constant and unremarkable as breathing, more constant actually, happening without pause across every tissue in the body from the moment a cell is viable until the moment it isn't. Proteins are the working components of biology, enzymes that drive chemical reactions, structural elements that give cells their shape, receptors that receive signals, channels that allow molecules to cross membranes, all of it protein. All of it being manufactured continuously, in quantities that stagger any attempt at intuition in every cell you have. Inside neurons, this manufacturing runs at a particularly high rate. Neurons are metabolically expensive cells, large, elaborately branched, maintaining electrical gradients across long distances, sustaining synaptic connections that require constant molecular upkeep. The protein turnover in a single neuron across a single day involves a number of individual molecules that has no useful comparison at the human scale. Most of those proteins do what they are supposed to do. The process by which a protein becomes functional is called folding. A protein begins as a linear chain, a sequence of amino acids strung together by ribosomes reading the instructions encoded in messenger RNA. That chain, as it emerges, is not yet a functional molecule. It is a string. What happens next, the process by which that string collapses and twists and folds into a precise three-dimensional structure, a structure determined partly by the sequence of amino acids itself and partly by the chemical environment of the cell, is protein folding. The structure is the function. A protein that has folded correctly into its proper shape will fit the molecular partners it is meant to engage with, will catalyze the reactions it is meant to catalyze, will perform whatever specific biological role it was made for. A protein that has not folded correctly, that has settled into a wrong shape, a shape that exposes surfaces that were meant to be buried, that brings together regions that were meant to stay apart, is a protein that cannot do its job. And depending on the protein, a misfolded version can do something worse than failing to function. It can be sticky. Misfolded proteins have a tendency, for reasons rooted in the physics of molecular surfaces, to aggregate, to clump together with other misfolded versions of themselves, accumulating into assemblies that the cell's normal quality control machinery struggles to dismantle. The cell has systems for dealing with misfolded proteins, molecular chaperones that attempt to refold them, proteasomes that break down irreparably misfolded ones, autophagy pathways that can engulf and digest larger aggregates. These systems work. They work continuously in every cell, and the vast majority of the time they are sufficient. The issue is not a single misfolded protein. The issue is accumulation over time, the slow grinding arithmetic of a clearance system that works at a certain rate against a production rate that, in an aging cell under metabolic stress, may exceed it. What escapes the quality control systems doesn't disappear. It accumulates. Two proteins have become particularly associated with this accumulation in the brain, and both appear in the research on sleep-dependent clearance with enough consistency to deserve naming clearly. The first is amyloid beta. Amyloid beta is a peptide, a short protein fragment, that is produced as a byproduct of normal cellular activity involving a larger protein called amyloid precursor protein, APP, which is expressed throughout the body but abundantly in neurons. A PP is cleaved by enzymes during normal cell signaling, and one of the cleavage products is amyloid beta, produced continuously, normally cleared continuously. Under healthy conditions, the production and clearance are roughly balanced, and amyloid beta concentrations in the brain's interstitial fluid remain low. When clearance is insufficient, whether because production increases, because the clearance systems are overwhelmed, or because the glymphatic flushing that carries amyloid beta out of the brain tissue is compromised, amyloid beta accumulates. In its accumulated form, it has a propensity to misfold and aggregate into oligomers, and then into the larger fibrillar plaques that appear in the brains of people with Alzheimer's disease. The link between amyloid beta plaques and Alzheimer's is complex and remains genuinely debated in the scientific literature. The amyloid hypothesis has dominated research for decades while also facing persistent challenges from researchers who point to the failure of amyloid-targeting drugs to reliably slow cognitive decline. The biology is not settled. The association is real. The argument about causation is ongoing. What is not in dispute? Amyloid beta is produced during waking neural activity, accumulates in the interstitial space, and is cleared more efficiently during sleep, when glymphatic flow increases and the expanded interstitial space allows fluid to move more deeply through the tissue. A floorboard settles somewhere below, a sound so ordinary it barely registers, the house adjusting its weight in the cold. Everything does this, adjusts, settles toward whatever equilibrium the night allows. The second protein is tau. Tau is normally a structural protein. It stabilizes microtubules, which are the internal scaffolding of neurons, the long cylindrical structures that give axons their shape and serve as tracks along which cellular cargo is transported. Tau bound correctly to microtubules is doing important work. It is in its proper form part of what allows a neuron to maintain its extraordinary length and structural integrity. Under certain conditions that include cellular stress, inflammation, and the presence of amyloid beta oligomers, tau becomes hyperphosphorylated. Phosphate groups are added to the protein at an abnormal rate, changing its chemical character, reducing its affinity for microtubules, causing it to detach and aggregate instead into what pathologists call neurofibrillary tangles. Tangles inside neurons, plaques in the spaces between them. These are the two hallmark pathological features of Alzheimer's disease, and both of the proteins that generate them are produced during normal neural activity and cleared, at least in part, during sleep. I want to be careful here about what this does and doesn't mean, because the connection between sleep and neurodegenerative disease is a place where the science is genuinely important and also genuinely easy to misread. The research, and there is now a substantial body of it, does show that sleep deprivation accelerates the accumulation of amyloid beta in the brain, that one night of disrupted sleep produces measurable increases in amyloid beta concentrations in cerebrospinal fluid, and that chronic sleep insufficiency is associated with elevated risk for Alzheimer's disease in epidemiological studies. These are real findings, replicated across multiple research groups, using multiple methodologies. What they do not mean is that insufficient sleep causes Alzheimer's disease in any simple, direct sense. The relationship between sleep, amyloid clearance, tau pathology, and dementia involves a number of interacting factors: genetic predispositions, vascular health, inflammation, metabolic state, and the causal direction is not always clear. It is also the case that Alzheimer's disease itself disrupts sleep, meaning the relationship may be bidirectional. Poor sleep promotes pathology, and the pathology promotes poorer sleep. This is mentioned not to complicate things unnecessarily, but because the science is complicated, and simplifying it into a clean warning, sleep enough or face consequences, flattens a biology that deserves its full texture. What sleep does each night is reduce the load, not eliminate it, not reverse pathology that has already developed, not protect against every risk or compensate for every vulnerability. But the clearance is real, the mechanism is established, and the cumulative effect of nightly glymphatic flushing across decades of life, carrying amyloid beta and tau and other metabolic debris outward through the tissue, through the perivascular drainage, into the cervical lymphatics, and from there into the body's broader waste processing systems, is not trivial. Each night some portion of what accumulated during the day is removed. Not all of it. Some, and some repeated across enough nights, adds up to something. The proteins that are fold wrong are not failures of the system. They are the system producing what any complex manufacturing process produces. Occasional errors, handled mostly but not perfectly by the quality controls that exist to catch them. The question over a life is whether the catching keeps pace with the producing. Sleep is part of the answer, not all of it. Part. In the tissue right now, amyloid beta is dissolving into the cerebrospinal fluid that has been pressing through the interstitial space across these hours of slow-wave sleep. The concentration in the extracellular fluid is lower than it was this afternoon. The microglia, in their ceaseless sampling, are encountering some of what remains and engulfing it. The astrocytes are taking up what their enzymes can process. The proteins that fold wrong are being cleared at the rate that the sleeping brain allows, which is faster than any other rate available. Which is why this matters. Which is why. The slow wave, when it arrives, carries something with it. Not metaphorically. There is an electrical event, large and sweeping, that moves across the cortex during deep sleep, and something about its passage, something about the coordinated neural activity it represents, appears to push the glymphatic system in ways that random neural firing during wakefulness does not. The wave and the clearing are connected. That connection is what the next chapter holds. There is an electrical event happening in your brain right now, not the diffuse, overlapping chatter of waking neural activity, not the fast, low-amplitude electrical fluctuations that characterize a brain attending to the world. Something slower than that, larger. A wave of synchronized electrical activity that rises across the cortex and then subsides and then rises again, with a period of roughly 1 second, sometimes faster, sometimes slower, but settling in the range that neuroscientists call delta, 1 to 4 cycles per second. You cannot feel it. Nothing about slow-wave sleep feels like anything from the inside. There is no subjective experience of the wave, no sensation of rising or falling, no awareness of the synchronized firing of millions of cortical neurons moving together in a pattern that, on an EEG, looks almost simple compared to the turbulent complexity of waking activity. The wave is there. It is large. It involves an enormous portion of the cortex in a coordinated electrical event that recurs steadily across the hours of deep sleep, and it appears to be doing something beyond what the EEG records. The slow oscillation, the technical term for what generates the delta wave, involves a cycling between two states at the level of individual neurons. During the upstate, neurons across a large cortical region depolarize together. They become electrically active, firing in a coordinated burst that, summed across millions of cells, produces the positive deflection visible on the EEG. Then almost simultaneously those same neurons enter the downstate. They hyperpolarize, their membrane potential drops below resting level, they go electrically short. The silence, summed across millions of cells, produces the negative deflection. Up, down, 1 cycle per second approximately, across the hours of slow-wave sleep. What determines the slow oscillation is not fully resolved, but it appears to emerge from the intrinsic properties of cortical neurons and their local connectivity, a property of the network rather than a signal arriving from outside the cortex. The thalamus, described in an earlier chapter as the sensory gating structure that closes down during sleep onset, also participates in slow-wave generation, its own rhythmic activity interacting with the cortical oscillation in ways that reinforce and shape it. The wave is, in this sense, the cortex speaking to itself, in a language that only exists in sleep. For a long time, the slow wave was understood primarily through what it is not. It is not waking activity. It is not REM sleep. It is not a stage of sleep associated with vivid dreaming, which is predominantly a REM phenomenon. Slow-wave sleep is the stage associated with the deepest behavioral unconsciousness, the hardest to wake from, the stage where a loud sound that would immediately rouse you from lighter sleep fails to register. It is the stage that the body prioritizes most aggressively after sleep deprivation. If you go without to sleep for a period and then sleep, the proportion of slow-wave sleep in the recovery night is markedly higher than normal. The body knows what it was missing. What it was missing was the wave. The connection between slow-wave activity and memory consolidation has been studied for decades, and the evidence for it is substantial. During the upstates of the slow oscillation, the hippocampus, the brain region most directly involved in the formation of new episodic and declarative memories, appears to replay the neural activity patterns generated during waking learning. Sleep spindles, the thalamic burst described in an earlier chapter, seem to coordinate with these hippocampal replays, and the coordination appears to facilitate the transfer of information from short-term hippocampal storage to longer-term cortical representation. Memory consolidation during slow-wave sleep is not a single process. It involves hippocampal replay, spindle-coordinated transfer, synaptic downscaling, and the pruning described in earlier chapters, all happening simultaneously, all interacting, producing by morning a brain in which the experiences of the previous day have been integrated into existing structure in ways that differ from how they were encoded during waking. This is understood reasonably well. What is less well understood, and what makes this chapter necessary rather than redundant, is what the slow wave does to the fluid. Somewhere in the middle of the house, a clock is doing what clocks do, not loudly, just persistently, its tick carrying through walls in the way that small regular sounds do when everything else has gone quiet, regular, unhurried, arriving and arriving. The connection between slow-wave sleep and glymphatic clearance was not part of the original description of the glymphatic system. It emerged later from work that asked a more specific question. Is it just sleep in general that drives the glymphatic surge? Or is there something about particular sleep stages, particular patterns of neural activity, that specifically promotes fluid movement through the tissue? The answer that emerged is that slow-wave sleep, and specifically the slow oscillation itself, appears to play a direct role in driving glymphatic flow. The proposed mechanism involves what the slow oscillation does to blood vessels. During the upstate of the slow oscillation, when millions of cortical neurons fire together, there is a coordinated demand for energy and oxygen, a local increase in metabolic activity that the vascular system responds to by dilating the blood vessels in the active region, increasing blood flow. During the downstate, firing stops, metabolic demand drops, vessels constrict again. The cycling of the oscillation therefore drives a cycling of vascular diameter, dilation and constriction, once per second across the hours of slow-wave sleep. The perivascular spaces, the channels along which cerebrospinal fluid enters the brain tissue, running alongside blood vessels as they dive into the parenchyma, are directly affected by changes in vessel diameter. When a blood vessel dilates, the perivascular space around it is compressed. When it constricts, the space opens. This rhythmic compression and opening may act as a pumping mechanism, pushing cerebrospinal fluid along the perivascular channels with each oscillatory cycle, driving it deeper into the tissue than simple pressure gradients alone would accomplish. The slow wave in this model is also a pump. Not the only pump. The cardiac pulsation and respiratory pressure changes described in an earlier chapter are also involved, but a pump that operates specifically during slow-wave sleep, at the frequency of the oscillation, with an amplitude that depends on how large and coordinated the slow waves are. The evidence for this mechanism is compelling but not yet conclusive. Most of the direct imaging work has been done in rodents, using techniques not yet fully translatable to human neuroscience. The perivascular spaces in human brains are smaller relative to total brain volume. The slow oscillation has somewhat different characteristics. and the relationship between vascular pulsation and cerebrospinal fluid movement in humans involves additional anatomical factors that the rodent models don't fully capture. What has been observed in humans is that cerebrospinal fluid movement, measurable by certain MRI techniques sensitive to flow, is coupled to slow wave activity. When slow waves are present, cerebrospinal fluid moves more. When slow waves are absent or suppressed, as happens with certain sleep medications or with the normal reduction in slow wave amplitude across aging, the cerebrospinal fluid movement is reduced. The coupling is real. The precise mechanism behind it is still. Laura Lewis's group at Boston University published work in 2019 using simultaneous EEG and fast fMRI to image cerebrospinal fluid flow in sleeping humans. What they found was that large, slow electrical waves in the brain were followed by large pulses of cerebrospinal fluid flowing into the brain from the spinal canal, the fluid rushing in as blood volume in the brain briefly dropped following the downstate, filling the space the receding blood had temporarily vacated. The wave, then the fluid. Every cycle, the brain's electrical activity and its fluid dynamics, locked together in a rhythm that runs through every hour of deep sleep, producing with each cycle a small increment of clearance, a small push of fresh cerebrospinal fluid into the tissue, a small displacement of interstitial fluid carrying its dissolved load outward toward the drainage. Small increments, all night. The slow wave is not just a signature of deep sleep. It is an active participant in what deep sleep accomplishes. The electrical event and the clearance event are coupled, one drives the other, and the other makes the former meaningful in a way that extends beyond neural consolidation into the basic metabolic maintenance of the tissue. This is what makes slow-wave sleep the most restorative stage. Not because it is the deepest, though it is. Not because it is the stage where dreaming is absent, though it mostly is, but because it is the stage where the wave and the fluid and the expanded interstitial space and the active microglia and the open aquaporin 4 channels all converge simultaneously, where every mechanism described across these chapters reaches its peak activity within the same hours of the same night. These slow wave coordinates what would otherwise be separate processes. It brings them into phase. Right now, if the wave is running, if you have crossed into slow wave sleep, or are near it, your cortex is cycling through up states and down states at its own slow tempo. Blood vessels in the active regions are dilating and constricting with each cycle. Cerebrospinal fluid is being pushed in small pulses along perivascular channels into the tissue. The interstitial space is open. The microglia are moving. The aquaporin 4 channels are full. The wave arrives. The fluid follows. Every second, approximately, it happens again. So ordinary at the scale of the sleeping brain that calling it remarkable feels like an imposition on something that has never needed to be called anything at all. It simply runs the way a current runs below everything, carrying what it carries in the direction it always goes for as long as the deep sleep holds. What the body does with what has been cleared, where the waste goes after it leaves the brain tissue, how the cervical lymphatics process it, what happens to the amyloid beta and the tau fragments and the adenosine and the ionic residue of a day's worth of thinking, that is a story that belongs mostly to the body and not to the brain. But there is one more thing that happens while the wave runs. In other tissue, in other systems, in the muscle and the gut lining and the skin, the dark is doing its own work. Not glymphatic, not neural, older in some ways, quieter. The brain has been the center of this story. That was appropriate. The glymphatic system, the suck wave, the microglia moving through neural tissue, these are brain processes, and the brain's relationship to sleep is extraordinary enough to hold attention for 11 chapters without exhausting the subject. But the brain is not the only organ that uses the night. The rest of the body is also working, quietly in parallel in ways that are less dramatic and less recently discovered, but no less real. The body's other tissues are using the hours of sleep to accomplish what the demands of wakefulness don't fully allow. Muscle is a good place to start, not because it is the most important, but because the repair it undergoes during sleep is perhaps the most intuitively accessible. During waking activity, and especially during any form of physical exertion, muscle fibers sustain small structural damage. Not injury. This is not the language of harm, but of normal use. The microscopic tears that occur in muscle fibers when they contract against resistance, the inflammation that follows, the protein degradation that creates the raw material for rebuilding. Exercise in this sense is controlled damage. The body tolerates it because the repair that follows leaves the tissue marginally stronger than it was before. The repair requires sleep. Not exclusively, muscle protein synthesis occurs during waking hours as well. But the hormonal environment of sleep, and particularly of slow-wave sleep, creates conditions that accelerate and deepen the rebuilding in ways that waking rest does not replicate. Growth hormone is released in pulses throughout sleep, with the largest and most sustained pulse typically occurring during the first bout of slow-wave sleep in the early part of the night. Growth hormone stimulates the uptake of amino acids into muscle cells and promotes protein synthesis, the literal rebuilding of the damaged fibers from the available molecular components. Without slow-wave sleep, growth hormone release is substantially reduced. The building materials arrive in smaller quantities. The repair is less complete. This connection between sleep and muscle repair is well enough established to have influenced how athletic training is understood. Recovery is not a passive absence of effort, but an active biological process that requires specific physiological conditions to proceed. Sleep is not the time when nothing is happening. It is the time when certain things happen that cannot happen any other way. Growth hormone has roles beyond muscle. It is involved in fat metabolism, immune function, bone maintenance, and cellular repair across multiple tissue types, a signal that the body uses to indicate broadly that resources are available for building and maintenance rather than immediate survival. Its pulse during slow-wave sleep is, in this light, something like a nightly maintenance signal. Conditions are right so the work can proceed. The gut does something different. The intestinal epithelium, the single layer of cells lining the interior of the small intestine and colon, is one of the most aggressively renewed tissues in the body. The cells of this layer, called enterocytes, live for roughly 3 to 5 days before being shed into the intestinal lumen and replaced by new cells migrating up from the crypts at the base of the epithelial folds. The turnover rate is extraordinary. The entire surface area of the intestinal epithelium, which is estimated at around 30 square meters when its microscopic folds are accounted for, is replaced roughly every week. Sleep appears to influence the timing of this renewal. The proliferation of the stem cells that generate new enterocytes, the mitotic activity in the intestinal crypts, follows a circadian pattern, with peak proliferation occurring during the rest phase. In humans, this means the gut cell renewal is biased toward the sleeping hours. The exact mechanisms involve circadian clock genes expressed within the gut tissue itself, responding to local signals that include feeding patterns, light exposure reaching the gut indirectly through systemic hormonal signals, and the broader physiological state that sleep creates. The gut is, in this sense, reading the same biological clock that the brain reads, and it is using the same window. Something about the gut doing this, renewing its own surface during sleep, quietly replacing the cells that spent the day in contact with everything passing through, feels appropriate in a way that is difficult to articulate without reaching for something that sounds more poetic than it is. It's just biology, time to the cycle, using the window, but the body's use of that window is, taken all together, not small. A faint smell of something cooling, the kitchen maybe, or just the particular smell that houses have when the heating drops and the air stills. It arrives and stays and becomes part of the room. The skin is the third system worth naming, though it could easily be the first. It is the body's largest organ, not an impressive claim until you consider what it does. Barrier against physical damage, microbial invasion, UV radiation, and water loss, all simultaneously across a surface area of roughly 2 square meters. Maintaining that barrier requires constant cellular replacement, and the outermost layers of skin, the stratum corneum, composed of flattened keratin-filled cells called corneocytes, are shed and replaced continuously. The rate of this replacement accelerates during sleep. Skin cell proliferation, the division of keratinocytes in the basal layer of the epidermis, follows a circadian pattern with peak mitotic activity occurring at night. Growth hormone, released during slow-wave sleep, drives some of this increase. The reduced blood flow to the surface during sleep, the drop in skin temperature, and the altered immune surveillance of the tissue all contribute to creating conditions that are, in some poorly understood but measurable way, more favorable for cellular repair and renewal. UV exposure during the day causes DNA damage in skin cells. The photoproducts that, if unrepaired, contribute to mutation and, over decades, to skin cancer. The repair of this damage, nucleotide excision repair, the cellular mechanism that finds and fixes UV-induced DNA lesions, is not exclusive to sleep, but it runs more efficiently in the absence of the cellular stresses that daytime activity and UV exposure impose. The night gives the skin a version of what the brain gets from the slow wave, not identical, not driven by the same mechanisms, but belonging to the same broad category of biological opportunity that darkness and sleep create. There is a word that appears in chronobiology, the study of biological time, that is useful here, Zeitgeber. It means literally time giver. It refers to external signals that entrain biological clocks, that synchronize the body's internal rhythms to the rhythms of the environment. Light is the primary Zeitgeber for most organisms, including humans. The body reads the presence and absence of light through photoreceptors in the retina that project to the suprachiasmatic nucleus in the hypothalamus and the brain's master circadian clock, and uses that information to time virtually everything, including the repair. Darkness tells the body it is time to rebuild, not through any single mechanism, but through the coordinated effect of melatonin rising, core body temperature dropping, growth hormone pulsing, cortisol falling, sympathetic nervous system activity receding, a constellation of physiological changes that together create the conditions under which the gut renews its surface, the skin repairs its DNA, the muscle rebuilds its fibers, and the brain runs its slow wave and flushes its waste. Darkness is not rest. Darkness is a signal, and the body, reading it, begins. The systems described in this chapter don't know about each other. The enterocytes dividing in the intestinal crypts are not communicating with the keratinocytes in the basal epidermis. The growth hormone signal reaches both, in a sense. It circulates in the blood and touches many tissues, but the specific repair processes in each organ proceed through local mechanisms, local clock genes, local cellular machinery that responds to local conditions. The coordination is systemic but not directed. It emerges, the way so much of biology does, from many separate things responding to the same signal at the same time and producing together something that looks like a plan. It isn't. It's just timing. But timing in biology is nearly everything. The brain is still running its slow wave. The microglia are still moving. The glymphatic fluid is still pressing through the expanded interstitial space. And at the same time, in tissue that will never generate a thought or carry a memory, the body's other systems are doing their own version of the same thing, using the night, using the dark, using the particular biological conditions that sleep creates to accomplish what the day, with all its demands, did not allow. The darkness is not empty. It never was. You didn't do any of this. That's the first thing worth sitting with, at this hour, after everything that has been described across the length of this night. The glymphatic surge, the slow wave, and the fluid it drives, the microglia extending and retracting through the tissue, the synaptic pruning and the protein clearance and the gut renewing its surface and the skin repairing the day's accumulated damage. None of it required your participation. None of it asked for attention or intention or effort. It simply ran the way it runs every night, in the tissue that produces everything you are, without once surfacing into the awareness that tissue generates. You were not present for it, and it happened anyway. There is something about this that takes a moment to fully receive, not because it is difficult to understand, but because it sits at an angle to how we tend to think about ourselves. We experience ourselves as agents. We do things. We decide and intend and act, and the body moves in response to those decisions. This is real. But it is a small slice of what the body is doing at any given moment. And during sleep it is not even that. The agency goes quiet, the intentions dissolve, and the biology continues without them, running processes of maintenance and repair and clearance that the waking, deciding self has no access to and no influence over. The body is competent in ways the self is not aware of. This is not a diminishment of anything. It is, if anything, the opposite. What has actually changed in the tissue during the hours of this night is a question that science can answer partially, honestly, and with appropriate humility about what remains uncertain. Amyloid beta concentrations in the interstitial fluid are lower than they were at the start of sleep. The studies that have measured this, using cerebrospinal fluid sampling in humans at different points across the sleep cycle, using microdialysis in animal models to track extracellular protein concentrations in real time, consistently show a drop in amyloid beta during sleep and a rise during wakefulness, a daily oscillation that reflects the balance between production and clearance across the cycle. The clearance that has been running tonight has shifted that balance toward lower concentrations, not to zero, not to some absolute clean state, lower. Synaptic weights across the cortex are, in at least some regions and by at least some measures, reduced from their end-of-day values. The dendritic spines that were pruned during the night are gone, not weakened, not modified, structurally gone, the postsynaptic specialization engulfed by microglia and broken down. The connections that remain are on average slightly scaled back from the potentiated state that a day of learning and experience had produced. The signal-to-noise ratio of the network, its ability to register new information above the background of existing connectivity, has been restored toward a level closer to what it was yesterday morning. The network is not the same network that fell asleep. In small but measurable ways, it is a slightly different one. The growth hormone that pulsed during early slow-wave sleep has moved through the blood and reached the muscle and the skin and the gut, delivering its maintenance signal to tissues that were waiting for exactly that signal. The intestinal crypts have divided. new enterocytes have begun their migration toward the epithelial surface. In the skin, nucleotide excision repair has been working through the UUV-induced DNA lesions that accumulated during the day's light exposure. Some of those lesions are gone, some remain and will be addressed in the nights that follow, or will not be fully addressed, in the ordinary imperfect way that biological maintenance always proceeds. Maintenance is not perfection. It is the sustained effort to keep the gap between current state and optimal state from widening faster than it can be closed. Something has settled in the room, not the temperature exactly, though perhaps that too, but something in the quality of the air, the particular stillness that arrives in the deepest part of the night when the sounds of the neighbourhood have fully ceased and even the house has finished its settling. The kind of quiet that doesn't press on you, that simply is. I want to say something about uncertainty here at the end of this particular night, because the science described across these chapters is real and it is substantial, and it is also, in several places, genuinely incomplete. The glymphatic system exists. Its role in waste clearance during sleep is established well enough to be considered foundational to how the field now understands sleep's function. But the precise mechanics of fluid movement, the exact contribution of specific sleep stages, the degree to which findings in rodents translate to human brains, these remain areas of active research with open questions. The synaptic homeostasis hypothesis is compelling and well supported, and also genuinely debated. The microglial pruning story is real, and also still being assembled. The connection between chronic sleep disruption and neurodegenerative risk is a real signal in the epidemiological data, and also not a simple causal story. Science that is honest about its own edges is more trustworthy, not less. What you've been listening to tonight is the field as it currently stands, not as settled fact to be memorized, but as a living account of what careful observation has found and what it hasn't yet found and what it suspects. The version of you that wakes from this sleep will be different in small ways from the version that fell into it. Not dramatically different, not transformed. The continuity of identity persists, the memories, the dispositions, the particular texture of personality that makes you recognizable to yourself. All of that carries across the sleep intact, and is in some cases more intact for having been processed and consolidated during the night. You will wake as yourself, but the tissue will have changed. The molecular environment will be shifted. The network will have been pruned and rescaled. The interstitial space will have closed back down as the slow wave recedes and the neurons begin firing in their waking patterns, and the potassium rises, and the cells draw their water back in. The microglia will have slowed their pruning and returned to their baseline surveillance. The glymphatic flow will throttle back toward its daytime rate. The waking state will reassemble itself as it always does from the biology that sleep leaves behind. If tonight was useful to you, if the slow pace of it, the way biology can be taken seriously without urgency, helped the day release a little, then what this channel is trying to do is working. It exists because there are people who find that science and sleep are not incompatible, that curiosity can be a path toward rest rather than away from it. Knowing that others are here for this, in the quiet of their own rooms, on their own versions of this night I tend to find it genuinely sustaining. If you want to keep this space going, membership is one way to do that. Super thanks is another. And simply being here, returning when the night asks for something slow matters more than either. Leaving a comment when you surface if something in tonight's journey stayed with you. I read those, all of them. And if this is something you'd want to return to, if the idea of a place where biology and sleep meet quietly is one you'd like to carry with you, Then subscribing keeps that door open. No pressure in that, just an open door. The slow wave, if it hasn't already, will return. The fluid will follow it. The microglia are still moving through the tissue, extending their processes into spaces that were dark before this channel existed, before any of this was described, before there were words for any of it. They were doing this when the first human slept under open sky, when there was no science of sleep, when the brain was entirely opaque to itself. They did not need to be understood to work. They are still working. Right now, in the tissue, in the dark, in the particular quiet of a body that has stopped asking anything of itself for a few hours, the clearing continues. The wave rises and the fluid follows, and the spaces between cells hold their nighttime width, and the proteins that fold wrong are carried outward, and the connections that should not last are gently, precisely, without drama or announcement, released. You are being maintained, without knowing it, without needing to. The night has been doing this work the whole time. It will keep doing it for as long as you let it.